The Department of Energy Rifle Subsurface Research Station was a former uranium mill processing site. Although the site has been reclaimed, residual uranium remains bound to the soil and sediments beneath it. That uranium can find its way into the adjacent Colorado River. Research done at the site since the early 2000s has focused on the ability to stimulate the activity of subsurface microorganisms that can convert that uranium from a form that's dissolved in groundwater, converting it to a form that's bound up or insoluble such that it does not want to flow toward the river system. This process is actually broadly relevant at a variety of similar sites to Rifle throughout the Colorado River Basin, where research is looking to find ways to minimize that flow of uranium from such sites to their adjacent river systems. Work done in partnership with the DOE Joint Genome Institute has been critically important for the success of the work that we've been pursuing at Rifle. What's been done has been very detailed sequencing of the genetic information of the subsurface microbial community that allows both for the identification of the organisms present as well as their functionality. In understanding their functionality with respect to a variety of critical interactions between elements of interest, carbon, nitrogen, and sulfur, as well as uranium, would not have been possible without this collaborative effort. Taking a look across a site like this, one can imagine collection of data that allows us to pinpoint areas of the subsurface that represent targets. These are also referred to as hotspots here. And our ability then to selectively identify and then probe those hotspots with the installation of wells results in the recovery of sediments and groundwater that constitute samples that can be sent to a broad network of collaborators, including those at the DOE Joint Genome Institute. Following preparation and extraction of genetic information from these sediments and groundwater, we're able to identify not just who's present there, but the functionality of those organisms associated with these samples. The research being done here at Rifle is really important not just for this site, but for a wide variety of similar sites throughout the Colorado River Basin. Specifically, the very comprehensive understanding of the interplay between water, microorganisms, and the long-term fate and transport of contaminants is critical to enable long-term decision-making for how to make use of former sites such as this.